Welcome everyone to the Water, Wetlands, and Watershed Seminar. It is fall 2020. It's September 30th. Today we have the pleasure of hearing from Dr. Natasha Woods. Before I introduce her, I'd like to just ask any announcements. Any clubs have any events coming up or any activities that you'd like to announce? I know we have fewer events than usual, but Dr. Clark, you have something?
Yeah, the Wetlands Club will be meeting next week. I do not have the link at this point, but I will send it out to at least the student list that we have for the class. So if you're interested in the Wetlands Club, it's a virtual meeting, so pretty much anybody is welcome. Hope to hear from them what we might be doing more physically. I think they had some discussions about canoe trip, but I'm not sure where that stands at this point. So even though I'm the advisor, I don't know much. So,
well, you're not big on Facebook, but there's the Facebook link to the UF Wetlands Club in the chat if you want to check that out. Any other announcements? All right, well then, I'd love to introduce Dr. Natasha Woods. Dr. Woods is an assistant professor of biological sciences at Moravian College in Bethlehem, Pennsylvania. She earned her PhD at The Ohio State University and received 2 postdoctoral fellowships, the Ford Foundation and an NSF fellowship for her postdoc work at Virginia Commonwealth University. And her research focuses on plant community dynamics in the coastal zone, particularly in maritime forests. We'll hear about that today. And she's broadly interested in the processes that cause ecosystems to assemble, reassemble, and disassemble. She's going to tell us what that means, but mostly focusing on how plants facilitate or compete with each other, how woody plants encroach, how seeds disperse and germinate in a very stressful coastal environment, and about the stability of coastal dune vegetation. So with that, Dr. Woods, the floor is yours. Please go ahead.
Thank you, David, for that great introduction to my work. Today, I will be talking about community disassembly and reassembly in maritime forests. So just to give you a little bit of background on maritime forests, they're coastal wooded habitats that are found on higher ground than dune areas, but still within range of salt spray. They're prevalent throughout the Atlantic and Gulf Coast in the United States and many other coastal communities around the globe. They represent a climax community. So succession typically happens in coastal areas from primary dune to grassland, to shrubland, and to climax maritime forest. And on barrier islands, maritime forests represent the most stable part of that island. To give you an outline of my talk for today, I'll be discussing forest disassembly on Hog Island and Parramore Island, and kind of contrasting maritime forest resilience on barrier islands versus the mainland. So if you can see my screen here, you'll see that these are Virginia barrier islands, and I'll be comparing resiliency of forest on barrier islands to those on the mainland. I guess you say that does not look like a mainland to me. Well, this is the Delmarva Peninsula, and the Delmarva Peninsula consists primarily of Delaware and the eastern shore of Maryland and Virginia. So it's actually a mainland. And as we consider how forests disassemble, it's also important to understand how exactly they reassemble. Because when they reassemble, a lot of times they are reassembling in an environment that's altered from the previous environment, which allows species that are better adapted to that new environment to potentially compete for recruitment space. So we'll be looking at that as well. Coastal plant communities are among the most vulnerable ecosystems to climate change. They are on the forefront of elevated temperatures, increased atmospheric CO2, increased storm frequency and intensity, and of course, sea level rise. The relative rise in sea level is almost 5.3 millimeters per year for the Virginia coast. Land subsidence has been observed since the 1940s, and in the southern Chesapeake Bay region, near where we are, they've been recorded at, land subsidence has been recorded at 1.1 to 4.8 millimeters per year, and subsidence continues even today. So the Virginia Barrier Islands actually consist of 14 islands, and you see 12 of them here. They're owned in part or totally by the Nature Conservancy. So most of these islands are uninhabited. Wallops Island has the NASA facility on it, so there is habitation of that island, and Fisherman's Island is where the Bay Bridge, Chesapeake Bay Bridge Tunnel actually connects. So this island is also inhabited. Now let's look at historical changes on Virginia Barrier Island. Historical changes was recorded by Fenster et al., and they were looking at changes in erosion and accretion of these islands from 1940 to 2006. So all the way from Wallops to south in Fisherman's Island, you see there's been more erosion on these islands than accretion. So lots of island area loss. So at Virginia Commonwealth University, I got a chance to work a lot on Hog Island as well as Parramore Island, the island just above it. We have lots of data on a lot of these islands. Zenert et al. Actually reported from 1984 to 2011 using Landsat data. They actually examined the distribution of woody species on these islands. So in 1984, this was the distribution on Hog Island of woody species. And in 2011, we see that the distribution of woody species greatly increased. I guess you say, I thought we were talking about the demise of maritime forests. Well, if you put boots on the ground, you would see that the expansion is not of tree species, but of a native shrub called Morella cerifera. So Morella cerifera has expanded. It expanded over 400% on this island. Hog Island used to be home to a large community, relatively small community. It's 30 families that used to reside on the south side of Hog Island in a town called Broadwater, Virginia. In the 1930s, it was hit by a hurricane, and since then it continued to get inundated. And as the community continued to get inundated, these families decided to move their homes to the mainland. And that was an excellent decision, because if you wanted to travel to Broadwater, Virginia today, just pack a little patience and a little scuba gear, because the current location of Broadwater, Virginia is now in the ocean. The Nature Conservancy describes these islands as living coastlines. They experience lots of erosion and accretion, and they roll over and transgress towards the mainland. But if accretion is occurring on the bay side, they roll over and progress towards the ocean. So they're constantly shifting. So when I say the most stable part of the island is typically maritime forest, it's because of all this shifting that's taking place. Maritime forests enhance the physical resilience to storms, and they provide short-term stability, right? So you have all of these tall trees that are buffering the mainland from the full force of storms. But after multiple disturbances, they tend to disassemble. Vegetative composition and topography affect coastline response to disturbance, because some of these species on the island are disturbance reinforcing, such as your grasses, and rollover can easily take place. Whereas shrubs, if you get an expansion of shrubs, the shrubs are disturbance resisting, and they resist the movement of sediment. Maritime forests are threatened by storm surge, sea level rise, and encroachment of more salt-tolerant species. So this picture here is of Parramore Island. I took this picture in 2018. But this area used to be totally tree species. It was total maritime forest. But now You can see that marsh has encroached. Invasive Phragmites that we know thrives in brackish water is there. Morella cerifera, which is a moderately salt-tolerant shrub, is there. And finally, we get a representative of trees in maritime forests, and that's loblolly pine in the background. So you can get a sense that this community has experienced a lot of vegetative change. And we argue that perhaps geographic position may account for differences in maritime forest resilience to disturbance. So barrier islands experience the full force of storms. So they get the highest levels of storm surge, wave energy, relative to the impact of storms from the on the bay side or the backside of those barrier islands. So as nor'easters come down from the north and hurricanes come up from the south, you're going to get the greatest impact on the forefront and less of an impact on the back. So our field sites are Savage Neck Nature Preserve, and it's managed by the Virginia Department of Recreation and Conservation Natural Heritage Site. And there are 86 hectares of maritime forests at Savage Neck. And 16 of these 8 hectares were sampled in 2003. And we resampled them in 2018. The initial sampling was a few months before Hurricane Isabel hit. Hurricane Isabel was one of the worst storms for the Virginia Barrier Islands because it was a rare storm in that it actually hit the Virginia coast in that coastal area. Typically, we don't have storms that come up that high. And Parramore Island, we have a long-term data set that was established first in 1997, and we resampled from it in 2018. Parramore Island is part of the Virginia Coast Long-Term Ecological Research Site. So we have a lot of data about how vegetation has changed over time. So here's a Landsat image showing woody vegetation versus bare versus grass versus marsh. And this is going to come in to be quite important later on in my talk. Storms are the primary disturbance as well as sea level rise, and they can work in concert. So storms can temporarily alter relative sea level rise and often result in saltwater intrusion that can cause forest species loss, like we see here on Parramore Island. The capacity of maritime forests to experience resistance or resilience to storms may also be due to intensity and frequency and duration of these disturbances. So we wanted to examine this a little bit more. And so we have a chart here that shows mean sea level rise from 1997 to 2018. And you can see how mean sea level rise has changed. And since 2003, there were 21 tropical storms and hurricanes that came within a 100-mile radius that had the potential to impact Savage Neck and Parramore Island. And since hurricanes don't typically make landfall in our area, you say, well, what's sort of the big deal? Well, even a hurricane or a nor'easter passing through can have substantial impact on the vegetation on these islands. So my research questions were, what is the relative extent to which forests on the mainland and barrier islands are resilient to change after long-term disturbance? And we determined that by examining the resiliency of species richness from the two time points and resiliency of diameter at breast height. Our second question was, to what extent are forests on barrier islands more vulnerable to disturbance relative to the mainland forest? And our third question was, what are the long-term shifts in community composition? In other words, are these communities moving towards secession or disassembly? Our hypothesis was that forest disassembly will be higher on barrier islands than the mainland due to their position in the landscape. On Savage Neck, the first survey was done in 2003, and in 2018, we used those same methods to resample 16 hectares of forest. So we used the point quarter distance method by Cottom and Curtis from 1956. Transects were positioned from the edge of the forest to the base of the inland dune, and transects were positioned approximately in an east-west orientation. And we set up quadrats every 10 meters along the transect, and we sampled any woody species per quadrat that was greater than 3 centimeters in diameter. And we did this all the way down until we made it from the edge to the base of the dune. We also measured DBH. We calculated importance values based on relative frequency, relative density, and relative basal area of the species. And to compare these communities, we use non-metric multidimensional scaling of species basal area and analyze those NMS data through multiple response permutation procedure to see if these communities were significantly different from the 2 time points. On Parramore Island, Richardson in 1997 set up circular permanent plots. And so we used that same method in 2018 to resample these plots. We resampled 10 plots that were 8 meters in diameter. We measured DBH, we calculated importance values, and we also used long-term structural changes as determined by Landsat TM5 satellite imagery. And we also did NMS and MRPP as well. So let's get down to some results. So question 1 was asking about resiliency and species richness. So at Savage Neck, we see that liquid amber Styrocyphala was the dominant species in 2003. But it was reduced in dominance by 2018. A cerebrum, on the other hand, increased in dominance, and Pinus tata increased in dominance as well. So the top 3 species for Savage Neck remained the same from 2003 to 2018. So these graphs are showing the top 6 species, and I'm just highlighting the top 3 dominant. So we see that there's a similar distribution for the 2 time periods for the top 3 dominant species, because the top 3 dominant species remain the same. Now, when we look at resiliency in DBH during the 15-year time period, we separated the species based on age class. So every 10 centimeters was a different age class. And as we can see from 2003, we have a number of young trees. And then the DBH class in 2018, we see that those young trees moved into older age classes. So the frequency of trees in the 30 to 80 centimeter age classes increased. A cerebrum and Pinus tata increased in basal area by over 30%. Liquid amber Styrocyphala individuals were reduced by 52%, but the trees that remained, as you can see, increased in basal area. Now let's move over to Parramore Island. So on Parramore Island, Juniperus virginiana was the dominant species in 1997, but it was severely reduced by 2018. But it was still remained within the top 6 dominant. Pinus tata and Persea paltris both decreased in dominance, but still remained in the top 3. The big story here is Morella cerifera. It was not in the top 3 of dominant species in 1997, but it became the dominant species by 2018. Species richness was reduced by 33% during the 21-year time period, and Morella cerifera increased by 156%. Now, I changed up the age class data just to reflect trees versus Morella cerifera. So in 1997, you see that trees were more prevalent than the shrub Morella cerifera at all age classes. And then by 2018, we see that Morella cerifera has really increased. So the frequency of trees in the 0 to 30 age classes decreased for trees. No trees were detected as being larger than 30 centimeters in DBH. And basal area was reduced to 16% of the 1997 value. Most of the basal area consisted, as you can see, of Morella cerifera. Question 2 was asking about the vulnerability of savage neck versus maritime forest on Parramore Island to disturbance. So on Barrier Island, erosion occurs on all sides. During the study timeframe, Parramore Island upland was reduced by 9%. And here we can see Parramore Island Landsat data from 1997 to 2016, but I'm going to blow this up on the next slide. Let's kind of focus on these pictures right here. Let's go on a field trip. If we were on Parramore Island and we got off the boat, the first thing we would come in contact with is ghost forests that would be in this bare area. Ghost forests are basically dead trees. And in this particular case, dead trees that are piled up on a beach. If we went a little further into this community, we would see transitioning grassland. So this area that used to be covered in trees is now covered in grass. It is transitioned into a grassland. And if we go a little further and finally make it to a maritime forest, we'll see that that maritime forest has been encroached with species that are more tolerant, salt tolerant than tree species are. So here we have Landsat image from 1997 to 2016. And the only thing we did was superimpose our study plots on top of the Landsat imagery data. So all of the white dots were the original plots from 1997. And all of the black dots are the plots that we resampled in 2018. And how about we take a view of where are they now? You know, they have the little shows that say, where are they now? Let's find out where some of these plots are now. So if we look at these plots here and compare them to where they are now, they're actually on the bare area. So they went from being maritime forest. So all of these white dots are in areas that was formerly maritime forest. They were part of that initial sampling of maritime forest. We see a whole swath that used to be woody species that's now on bare land. So it'd be on the beach. And then we come to more plots that used to be woody, but now they are bare. We go down even further, we see woody species that used to be woody in areas that are now bare. You can probably see where I'm going with this. So from satellite imagery and ground truth, we know that 33% of the plots that were originally in that 1997 set are now in the ocean. 34% of the plots have converted to grassland or marshland, and 33% of the plots remain dominated by woody vegetation. But we see that that woody vegetation is increasingly becoming Morella cerifera. Whereas at Savage Neck, all plots surveyed in 2003 remain forested in 2018. Question 3 was looking at more of the long-term shifts in community composition from the two time periods, and we did NMS based on species basal area. So the black dots represent plots from 2003, so what the composition looked like in 2003. In 2018, sort of what the composition looked like in 2018. There's a lot of overlap. I'm not for sure if you can see that there's an overlay here, but there's quite a bit of overlap. Even though these communities, according to MRPP, are significantly different from the two time frames, there's quite a bit of overlap relative to Parramore Island. In Parramore Island, the 1997 distributions are in black dots and white dots for the 2018 distribution. And you can see that the 2018 species distributions or compositions are mainly clustered together. So definitely two totally different trajectories for these two maritime forest communities. So in conclusion, we see that maritime forest at Savage Neck is more resilient to disturbance than forest on Parramore Island. And Savage Neck appears to be undergoing a typical successional pattern. And on Parramore Island, areas closest to the shoreline have been completely lost to erosion or converted to grassy areas. And of the remaining interior forests, there is some form of resilience in conversion to shrub dominance because it still maintains a woody state. Not all communities may recover to their pre-disturbance state. The forest on Parramore Island is vulnerable to upland area loss and conversion to different vegetative communities, which we saw clearly. But it's unclear if a threshold has been reached on Parramore Island that would implicate a tipping point to continual disassembly of the maritime forest community. So now it's time to think about regeneration. How do these communities that have been so, or this community that has been so severely disturbed, how does it come back? How does it reassemble? So community reassembly depends on the extent to which tree species can regenerate in the altered environment. And for these barrier islands that are getting encroached by Morella cerifera, the extent to which Morella cerifera regenerates in the altered environment. So what do we know about Morella cerifera? Well, its expansion alters island migration, leading to decreased island area. It forms a symbiotic relationship with a nitrogen fixer that allows it to flourish in an area that's relatively nutrient poor. A lot of this is sandy beach. It alters microclimate, which favors its own establishment because it forms these really shady thickets that shade intolerant species cannot flourish in. Within 15 years, a grass dominated swell can become a closed canopy thicket. So here we have a young thicket. So these Morella growing in a grassland, but within a few years, it's going to be fully thicketized. The question that we want to ask is, does Morella cerifera have an advantage over native species when salinity is high? And I'm always interested in early stage development. So to answer this question regarding reassembly, we germinated seeds of Morella cerifera and common coastal tree species at increasing salinities. So the tree species that we want to include in this study are species that are common along the Atlantic coastal plain. So far, we have stratified and completed experiments for 5 of these species. So hackberry, Celtis occidentalis, sweet gum, liquid amber styrosiphylla, red maple, which is Acer rubrum, loblolly pine, which is Pinus tata, and red bay, which is Persea bourbonia. A number of these species you probably have right where you are in Florida. So our methods was to moist cold stratify seeds. We treated them to 5 salinity, 0, 2, 5, 10, and 20 parts per 1,000. Treatments were added every 5 days. There were 4 replicates per treatment, 25 seeds per replicate, and the duration of the experiment was 55 days. And this was a growth chamber experiment with a 14-hour photo period. The indices at which we measured germination was total germination, the number of seeds germinated over the course of the experiment, mean germination time, and it's to calculate mean germination time is actually the measure of the spread of total germination normalized by the number of seeds that germinated per experiment. You can think about it as the time to reach 50% germination. Mean germination rate is the number of individuals that germinated divided by the time it takes for germination to occur. And the reason why we look at this indice is it speaks to a delay in germination. Was germination delayed by an increase in salinity? And then finally, we look at the Kaplan-Meier survival curve. And that's the probability of not germinating at all. So this is typically used by medical researchers to determine the probability that a subject will not survive given the certain risk of a certain treatment. And ecologists kind of put a different spin on that. And we say, hey, what's the probability given this particular disturbance, this seed will not germinate? So we see here total germination. The first thing that may pop out to you is that Acer rubrum was not affected at all by the different levels of salinity. But the spoiler alert here is that studies show that it's quite sensitive at the seedling stage of development to salinity. So don't get too happy about Acer rubrum just yet. So at 0 and 2 parts per 1,000, there was no difference in germination from the control. But by 5 parts per 1,000, you start seeing significant differences in total germination relative to the control. For liquid amber styrosiphylla and Celtis occidentalis, germination significantly decreased at 5 parts per 1,000. And then at 10 parts per 1,000, all of the species that were affected by salinity were affected by that 10 parts per 1,000. But I would in particular like to point out that Pinus tata, even though its germination was significantly reduced, its germination was still higher than all of these species here on the bottom. So even with reduced germination, it can still thrive in that environment relative to the selected species in this study. And then by 20 parts per 1,000, a lot of these species didn't germinate at all. Pinus tata and Acer rubrum were the only species to germinate. And just to give you a reference point, ocean strength water is like 33 to 35 parts per 1,000. So a number of these species will not even be germinating at 20 parts per 1,000. That's something to note. So now when we look at mean germination time, so remember, it's more like the time to reach 50% germination. Acer rubrum, it's not affected. Persea borbonia took the longest time to reach mean germination time. All of these were affected on some level. It's almost like a little stair step here. As salinity increased, so did the time to reach mean germination time. So it was significant for at 10 parts per thousand for 3 of the species, and at 20 parts per thousand, you know the story already. Only Pinus Tata and Acer rubrum were responding anyway. All right. Now, when we look at
Natasha, we lost you. You got your mute on somehow.
Oh, okay. So where did you lose me at?
Just when you came to this slide.
Okay, just when I came to this, you know what? I was trying to move my bar across the top. I do this in lecture as well. It's like I'm moving my bar. Okay, so mean rate of germination. Remember, that tells us about a delay in germination. So it's about the speed of germination relative, and also takes into account how many actually germinated. So the ones with the highest rates of germination was the Acer rubrum, Pinus Tata, and Celtis occidentalis. Persea borbonia didn't germinate very well. Its germination was low, but as you can see, its germination was not affected at all by salinity. There was no delay in germination experienced for Persea borbonia at all. Whereas Pinus Tata, Persea borbonia, and Celtis occidentalis, they all experienced the delay in germination, and this is on the wrong slide right here. And by the time we got to 10 parts per thousand, it took longer for these species to germinate. So they're responding to the salinity. And then when we get to the end, we're looking at the probability of not germinating. So probability of not germinating over the time it took for, over the course of the experiment. So Acer rubrum, of course, probability of not germinating for this species is very low. For 0 and 2 parts per thousand, there's a relatively low probability that these species won't germinate. I only started at 40 because I got a nice spread for Persea borbonia at 40 because it took so long to germinate. So there's no significance about day 40. And by the time we reach 5 parts per thousand, 3 of the species have a significantly increased chance of not germinating at all. And all of the species that were responding to treatment, all of them have an increased probability of not germinating at 10 parts per thousand. And by 20 parts per thousand, there is a 100% probability that all the species in the yellow boxes will not germinate. So what did we learn from this? This study showed that Morella sorifera does not appear to have an advantage at the germination stage of development over the tree species in this study. Its total germination was equally affected by salinity as Persea borbonia, Celtis occidentalis, and reduced relative to Acer rubrum and Pinus Tata. Morella sorifera showed increased mean time to germination and experienced a delay in germination with increased salinity that was similar to many of the tree species in this study. So it really didn't do any better. But what's the ecological significance? Why analyze germination so many different ways? Well, a delay in germination may change the competition outcomes and change the trajectory of the community. Pinus taeta can withstand a delay in germination at higher salinities because it started out with a higher germination. An increase in soil salinity will adversely impact the germination of maritime forest tree species, in particular for those that start with low germination. And when trees succumb to salt intrusion, regeneration from seed will be difficult for some species at higher salinities. And a spoiler alert here, Morella cerifera is also clonal. So it does not have to germinate via seed. So which may contribute to its capacity to expand so greatly. So just a quick plug for my future experiments. I want to continue on with the germination and look at more species and how they are affected by salinity, and take it to the next stage of development and see how they respond at the seedling stage of development, but also examine Morella encroachment into grasslands. So the shrub Morella cerifera is not just encroaching into maritime forests, but also into grasslands. And that could be, there's studies that suggest that grasses facilitate shrubs. So that may be a possibility. And the fact that there's a primary dune that may be protecting it, the elevation of those dunes differ, and Morella cerifera may be responding to that. And I won't go any further about that. With that, I would like to thank the Coastal Plant Ecology Lab, the Ford Foundation, the VCR, LTR, who funded my postdoc, and VCU for all the support that I received while I was there. And this is Julie Zennert, who is in charge of the Coastal Plant Community Lab. So I really give a lot of thanks to her and the entire team. Austin, who also did a lot of work. This was part of his undergraduate project. So with that, I will take any and all questions. And I started out with a really rough picture, so I wanted to give you something beautiful to look at the end.
Thank you, Natasha. That's great. Really appreciate seeing it sort of from both the outcome, the actual forest change, and then, you know, thinking about from life cycle, seeing the seeds sort of at the end as an explanatory sort of piece of the puzzle. So we'll open it up to questions. If you're in the Zoom room, please feel free to unmute yourself and ask any questions. And then we'll convey anything coming in from the YouTube via the chat. So any students or faculty in the class have a question?
Hi, Natasha. That was a wonderful presentation. Just your overall field trip was a great orientation to the island and all the work you've been doing. I've got a question regarding the expansion of the Morella cerifera as it relates to the invasiveness of Phragmites. And do you see competition along those fronts? Do you see that the Morella can actually get established within a Phragmites stand, or is that, I mean, if we think about competition for light, are those two competing for that critical resource? I'm just kind of curious, do we see a native species potentially outcompeting the Phragmites, which I'm assuming you would categorize as one of your grassland type species? So
that's actually an excellent question. And we actually haven't looked at that, but just thinking about Parramore Island, there are definitely areas that are covered with just Phragmites and no Morella is present whatsoever. But then you have other areas where you have lots of encroachment of both these species, like I showed in the one picture where you have one level of Phragmites and then another level of Morella. And it may be an age thing, who arrived first. It may be a founder's effect, right? So if Morella got a foothold in first, and then maybe Phragmites came, and then the marsh, and they're all competing now for this limited amount of space.
I suspect there may be some elevation critical thresholds for hydrology and the Phragmites versus where the Morella is. So there may be another dimension in that partitioning within your grassland. But yeah, I'll be curious to see what you find in the future if you start looking at that kind of spatial distribution. Yeah.
That's a good direction to go in. Thank you, Mark.
Sure.
I have a question. Just it's a kind of from a selfish point of view. I'm working with my former PhD student who's now a postdoc at UVA, Elliot White, and we have a study that looks in a similar way at a couple of sites with a long time span in between, and we characterize the plant community composition. So focused on the canopy species in a forest undergoing saltwater intrusion. So very, very kind of similar. And one thing we're kind of facing a struggle with is when you have a sample and a resample event, like you have in your the neck, the one on the inside and the one on the outside, right? And you compare those 2 times, and you can say like, well, I saw 200% increase in Morella, or I saw 50% decline in Acer, for example. But we have a hard time saying how that's statistically significant or not, because the unit is almost your sites were your unit, right? And so so how did you approach that? I saw you had an NMDS or a PCA, some kind of ordination. How did you approach the statistics for that kind of experimental setup?
That's a great question, because Julie Zenner did the statistics on that. But so you can definitely contact Julie about how she did the NMS. But what question are you getting at specifically?
I mean, you might say, like, for example, that, species X increased by 25%, but the unit of analysis is like, in your case,
it's a landscape. Yeah.
As opposed, to like, I mean, did you treat, I can ask Julie too, but do you treat the plots or the transects, like the points on the transects, as independent? We have some transects, but we don't feel comfortable making them independent measurements necessarily. And then doing some kind of distribution. It's a whole transect, like your case was a whole sort of, this group over here increased or decreased. So I was just wondering about that, but we can check.
Yeah, I definitely can see where you're going with that, but I couldn't speak to how exactly she did the analyses.
Okay, well, actually, I see Elliot was on YouTube and he has a different question for you. He asked, how does the interaction of salinity and shade tolerance impact growth and survivability of species? So
yeah, I didn't examine shade tolerance, but I think that's a very good question, because Morella is putting out these very dense canopies. And so any species even attempting to grow in the shade of Morella is also having to deal with that level of salinity as well. So yeah, that'd be something interesting to look at as well, a different direction to go with that.
That's also in his manuscript we're trying to get out with Elliot too. So I think he's got a very pointed question for you there. So I just generally thinking about this idea of, and maybe it's, too conceptual or just kind of navel gazy, but when you contrast disassembly and succession, can you tell us a little bit about that? I mean, in this very highly disturbed system where like succession is punctuated by disturbances all the time, and it's this kind of process, when you set up the mainframe, it was like, are you seeing disassembly or are you seeing succession? And I guess just as like an ecology lesson for the students, like how do you see those as being different?
So they're different in that when we think about succession, we think about a successional pattern, right? We think about this area is becoming more favorable for a more complex species and a more complex species, so we get to some type of climax, right? But then when we look at these communities that are highly disturbed, and in particular talking about Parramore Island, we see that succession is interrupted. The way we typically think about succession is interrupted in that, if I can go back to one slide, in that we see now shrubs going in 2 directions instead of 1 direction. So we altered this pattern, or I should say Morella cerifera has altered a typical successional pattern by being able to go backwards and forwards. That means it's able to inhabit 2 different types of environments, and how it's doing that, the mechanism behind that may be totally different for each perspective. So grasslands, it may be that the grasses are facilitating its establishment. And for encroachment into maritime forests, the mechanism may be totally different. It may just simply be that it can tolerate a higher salinity tolerance, and therefore it can go. And another aspect of this, I keep going, and another thing, and another aspect of this is the minimum temperature has increased. So Morella cerifera used to die back in the winter. But since we don't hit those minimum temperatures anymore, instead of dying back, it doesn't. Its growing season has increased. And so that's changing a typical successional pattern that we think of as being one way in one direction.
Thanks. Yeah, that's interesting. And that it just makes me think also further south, we have the same, higher minimum temperatures and mangrove encroachment also into the same sort of maritime, coastal forest setting. So that might be a fun comparative study to try to write about.
Definitely.
Andrew, do you have a question?
Yeah, Natasha, that was a nice talk. It's really interesting to learn about the natural history of your system. You seem to have a good grasp of that. And, as a kind of foundation for your, ecological conceptual questions. But I did have a question, something I just noted. I think it may have been Parramore Island when you were showing just the classification maps, basically how the islands look like they'd changed between, I think it was 97 and more recently. It looked like there had been some sort of a cut that went through the island, all the way from the seaward side to the leeward side.
Let's get us back there.
Yeah.
Right here.
Yeah. So right on the lower left-hand corner of the yellow triangle 97, you see a cut that goes all the way through the island, it looks like, or at least from the beach into near to the other side.
Is this the cut that you see? Oh, am I still? Okay, that one. Okay.
Yep. And in 2016, it looks like it's large. It's closed on the beach side, and certainly there's closure on what may be marsh. Yeah, marsh. I was just wondering. How much vegetation may have helped drive that closure, and also whether that if the closure is real and not just an artifact of some kind of imagery, but if vegetation may have driven that closure, and or if the newly closed area is a opportunity to look at some successional patterns on what's essentially new terrain at the bottom of that island.
That's actually a great question. So this is primarily marsh habitat here, and you're right, this is not an artifact. This is actually a closure of the island. So marsh, they're grasses, so they are considered disturbance reinforcing, so they can actually roll over and capture the increased sediment. It is a great opportunity to actually look more at this. So I'm focused on Hog Island and looking at how Morella is experiencing and changing based on dune elevation. And I'm looking at it on Hog Island because it's accreting a lot on one end. And so we can actually capture as Morella starts to encroach and as these elevations in dunes start to change. How exactly does this encroachment happen? What are the factors? And it's always good to do like this, catch something as it's happening or as it's happened, to actually be able to go in and see what exactly is driving it. Because I just my personal opinion, I think it's the vegetation itself that's actually closing that gap. But yeah, it's not an artifact, that's actually real.
And that closure, it sounds like it's mostly converted from essentially open water to marsh then?
Yes.
At least so far, okay. Yeah, okay, interesting. All right, thank you.
Oh, thank you, Andrew, that was a great question.
All right, thanks everyone. Well, we're a little bit over already, so let's just you can either unmute or use the reaction to give a round of applause to Dr. Woods. So thank you very much, Dr. Woods, for joining us. We appreciate it, and wish you the best of luck as you continue with the semester.
Thank you. You guys too. Stay safe down there.